Welcome to Punnett Square 101. Today we will learn how to make and use Punnett squares for monohybrid crosses. Gregor Mendel is considered to be the father of genetics. His experiments on pea plants in the mid-1800s helped form our basic understanding of the laws of inheritance. Supported by his fascination with Mendel's work, Reginald C. Punnett devised the Punnett square in the early 1900s. The Punnett square was a simple method to predict the number and variety of genetic combinations possible through a breeding event. Let's review some basic vocabulary. Monohybrid crosses represent Punnett squares that only explore one trait at a time. For example, eye color. The illustration at the bottom right is an example of a heterozygous individual. Heterozygotes have two different forms of a trait. For example, a copy of the blue eye allele from mom and the brown eye allele from dad. We symbolize that with a capital letter and a lowercase letter written as big B, little b, always keeping the uppercase letter first. Homozygous individuals have two copies of the same allele. In our example, brown eyes are dominant to blue eyes. Homozygous recessive individuals have two copies of the blue eye gene, one from mom and one from dad. We write that as a lowercase b, lowercase b. Homozygous dominant individuals would have an uppercase b, and this would present itself as blue eyes, as brown is dominant to blue. Here's our problem. Predict the results of a cross between a homozygous recessive female and a heterozygous male. After you read the problem, underline the important terms like homozygous recessive and heterozygous. You can also write out what those genotypes look like. For example, homozygous recessive would be little b, and our heterozygous male would have a genotype of big B, little b. Here is our monohybrid Punnett square. We'll start by writing out the genotypes at the top. For this problem, we need a heterozygous individual, big B, little b, crossed with a homozygous recessive, little b. Remember, in meiosis, our chromosome numbers are cut in half and they independently assort. The way that we show that on the Punnett square is by splitting up the copies we get for each gene. On one side of the square, we'll place mom's genetic information, little b. On the other, we'll place dad's information, big B, little b. Make sure to separate your letters. This represents how our reproductive cells only get one copy of each trait. In mom's case, a little b would go into each one of her eggs, and in dad's case, half of the sperm would have a big B, half of them would have a little b. Now let's figure out what's going to happen with our offspring. We can start by filling in the box at the top left. We would pull the big B down from the top and the little b from the side, so in the first box I have big B, little b. The uppercase letter should be written first, and this represents a heterozygous individual who would have brown eyes because brown eyes are dominant to blue. The box at the top right would say little b. At the bottom left, we would get big B, little b. And at the bottom right is little b. So what does this all mean? It does not mean that these parents are guaranteed to have 4 children. The Punnett square gives us the probability of inheritance of a particular trait each time the parents reproduce. If we look at the genotypes or the letter combinations in each box, there is a 2/4 or 1/2 chance that the offspring will be big B, little b, which means they will have brown eyes. There is also a 2/4 or 1/2 chance that they will be little b, or have blue eyes. So there is a 50% chance of having offspring with blue eyes and a 50% chance of having offspring with brown eyes each time these parents reproduce. Please try out the practice problems on Google Classroom. I look forward to moving into dihybrid crosses with you, and I hope this video was helpful.